So welcome back, everyone. It's my great pleasure to have you all back for year 3 now. We start 2022 Breakfast and Science seminars. There's been lots of changes in the CIBMs and lots of great news. And one in particular is the joining of Valentina Garibotto, who will be the CIBM PET UNIGE section head. And there are other news coming up as well, but I will save these for the end. For now, I'd like to thank Francois for chairing this, for introducing Valentina, and pass it on to him.
Okay, thank you. Just for the people who doesn't know her, I hope you see my presentation. At the UNIGE, Valentina is well known. She has been around in the PET and nuclear medicine arena since a long time. And for the people who doesn't know her, I would like just to share with you some of her itinerary. She's from Italy, not very far from Geneva. So she has got her MD degree in Genoa, and she had a specialty in nuclear medicine with equivalent of FMH in Milano, in Italy still. And after she moved to Germany, to UNIGE, well-known research center in MRI and also in nuclear medicine. And in 2009, she joined the HUG as intern in nuclear medicine. And she has got a research grant, and she started to do research in the same unit. And in 2013, she became what we call in French or in Geneva, chef de clinique, or attending in the same service. And in 2014, she has got a private docent in Geneva. And in 2020, so 2 years ago, she has got a title of professorship of associate professor of radiology. And since last year, she's a chair of nuclear medicine ad interim after the departure of Martin Walter. She has an impressive track record with more than 180 publications. She was participated in 6 FNS grants, and she's actually on a grant project on Alzheimer's disease, which is in fact her main clinical interest, all the degenerative disease and especially the Alzheimer. And she's also a Congress chair of the European Association of Nuclear Medicine, which in fact is a pretty important medical association. So without taking the floor longer, I will stop here my presentation, and I will let Valentina speaking about her research, and we are very happy to listen to her. So I let her microphone and screen sharing to her. Thank you, Valentina.
Thank you, Francois, for the very kind words, for the nice introduction. I will then share my screen. Let's see, you should see my presentation now, and possibly yes. Now in presentation mode, but I'll start from the beginning, which is possibly wiser. So now it should work, right? You see the title.
Yes.
Okay. So what I want to share with you today is how, in my opinion, of course, molecular imaging has changed our understanding of dementia. And so what I suggest as outline would be to discuss briefly about what I refer to as molecular imaging, Alzheimer's disease, and the link between the two topics, how molecular imaging biomarkers have impacted also Alzheimer's disease drug development, And one of the topics we've worked a lot is the molecular imaging biomarker validation process, how this ultimately resembles the validation of a drug, and what are the implications of it, to draw then together some conclusions. So first, when I refer to molecular imaging, I mean, it's probably possibly my bias. I think about nuclear medicine, but it's true that nuclear medicine is probably one of the, let's say, of the few or the only technique that allows seeing molecules in clinical practice. The main modality we use clinically is positron emission tomography. SPECT or single photon emission tomography would be very similar. Our systems, let's say, record the radiation that is emitted by the radiopharmaceuticals, by the tracers that we administer. And so the specific characteristics of the molecular imaging that we can do is driven by the tracers. These traces have, let's say, either the ability of being processed by a metabolic pathway, or they're specific for one molecule, one target molecule, and then they allow then building very different images. If you inject, for example, fluorodeoxyglucose, as in this case, so a fluoride attached to the glucose ring, you will get an image of the glucose metabolism. If you use a tracer that is targeting, that has affinity for amyloid plaques, you will have an amyloid PET. And if you have a tracer that is specific for tau aggregates in the brain, you will have an image which looks, again, different from the other, that shows you the presence or the absence of tau aggregates. So really the characteristic, the core element in our molecular imaging in PET are the radiopharmaceuticals. And this is why I want to mention briefly how these radiopharmaceuticals are produced. These are produced via medical cyclotrons. So these different molecules are obtained having, of course, chemicals to start with, and then a cyclotron is a structure that looks like this. You see, it's the size of a couple persons working in it here. Of course, it needs a large bunker and relevant shielding, because it has the result of its work is radioactivity. It accelerates a particle beam in order to produce this radioactivity. In the most commonly used is fluorine-18. And then with this fluoride-18, when you have the radioactivity, this is moved to synthesis modules in the laboratory, which allow binding the radioactivity to the molecule you want then to have the radiopharmaceuticals to then be injected and imaged. The synthesis is really a process that is very similar to the process of the synthesis of whatever pharmaceutical. So it's the production of a drug, has then the same, let's say, limitation and implications of producing vials, of making quality controls, and then shipping. The specific characteristic of radiopharmaceuticals is their very short shelf life. So radiopharmaceuticals are produced, and then in the case of fluorine-18, which has a half-life of 2 hours, they can be valid for up to 10 hours. So you have to imagine a drug that is produced specifically for the patient that will use it on that day. A few tens, when the yield is high, it can be as low as 3 or 4 patients per synthesis, which of course means a lot of work, and explains partly why the costs of this technique are quite high. Clarified what I refer to as molecular imaging in this talk. Let's briefly talk about Alzheimer's disease. You possibly all know that Alzheimer's disease takes its name from the first describer of its neuropathology. It's a psychiatrist, Alois Alzheimer, which at the beginning of the 20th century described the case of a lady having loss of memory, loss of autonomy, and that he could follow until death. And it's mainly on the brain tissue of the person that he could describe aggregates that he wasn't used to see in normal brains, that these are his original drawing, aggregates that then have been renamed and classified as amyloid beta plaques, so these plaques, extracellular plaques, and intracellular aggregates, neurofibrillary tangles that have been discovered to be composed by tau protein, a microtubule associated protein. Since this first, let's say, description of the case, both clinically and neuropathologically, there has been a long silence and long, in a way, lack of interest in the Alzheimer's disease condition until 1984, when the first clinical criteria have been proposed. And these, this international working group proposed criteria that were, clinically based. So describing the typical clinical presentation with a memory involvement in the, let's say, in the first phase of a series of patients. Alzheimer's disease is the most common cause of loss of memory and ultimately dementia in the age population. These criteria were only based on clinical characteristics and multiple series, then describing these patients that were described clinically and then following them until autopsy have shown that these criteria perform quite poorly diagnostically. So the sensitivity and specificity of these criteria is in the range of the 70–75%. So there are some cases that present with atypical symptoms that these criteria cannot capture, and the other way around. There are patients that present with a similar clinical picture, but ultimately do not have the pathology. And this occurs in about 20–25% of the cases. And then again, from these clinical description, let's say from this diagnostic criteria, so the first criteria that could be used clinically then to give a diagnosis, give an answer to the patients when they were presenting with these signs, there has been again a quite long time, over 10, almost 30 years, to have a novel suggestion in 2017. But then, as you see, it has accelerated. I didn't mention all the novel criteria here, 2017, then 2011, then 2018. And the acceleration in this phase is, in my opinion, not only mine, driven by the advent of specific markers, and in particular, molecular imaging markers. In 2004, we have the first clinical series of amyloid PET scans. So we have, for the first time, the ability of doing molecular imaging that visualizes these amyloid aggregates that Alzheimer's described. And then the 2007 International Working Group and then the 2011 National Institute of Aging criteria integrate this information into the clinical diagnostic criteria. So the diagnosis is not only based from 2007 on the, let's say, typical clinical picture, but it's typical picture plus amyloid markers. And then in 2013, we have the advent of tau PET. So the first radiopharmaceuticals that are specific for the tau aggregates, for the tau intracellular aggregates that Alzheimer described. And then in 2018, the National Institute of Aging has proposed a research framework in which they try to, they suggest, at least in research, not yet into clinical practice, to classify individuals on the basis of the presence of amyloid and tau, as Alzheimer's did, but in this case, not on neuropathology, not at autopsy, but in vivo. So in these criteria, I won't go into the details, but the biomarkers, so these measures that can be performed in vivo, that include mainly molecular imaging, are classified by their ability to see amyloid, tau, and the presence of neurodegeneration, which is considered a downstream effect of amyloid and tau aggregates. And then in this set, if you have amyloid and tau, irrespective of the clinical presentation, so a 180 degrees shift from the criteria of 1984, irrespective of the clinical presentation, if you have amyloid and tau, you have Alzheimer's. The reason for this transition this transition has also changed our way to consider the disease. And this is true for Alzheimer's disease, but it's also in a way generally true for what's considered all neurodegenerative conditions. I'm thinking about other situations associated with dementia, for example, frontotemporal lobe degeneration, or Parkinson's disease, for example. The current understanding of these diseases is that they evolve over decades. So you see on the, let's say, on the horizontal line, the transition in different phases and the evolution over time. And what the community has realized is that when you see the patient with its clinical manifestation, so dementia, loss of autonomy, cognitive impairment, you're seeing the end stage of a disease, which accumulated over years. If what happens in the brain, and already in the preclinical phase, when the person is fully intact in a way, normally functioning, is the accumulation of these pathological proteins, then the debate is still there if it's amyloid or tau, which accumulates first, if they're the cause or rather the, let's say, a sign of a process associated with the process. But I mean, there is agreement on the fact that both presence are can be detected long before their clinical symptoms. And this, of course, opens, I mean, a fully novel perspective, because if we describe a condition only post-mortem, pathologically, of course, there is not much, let's say, space for intervention. But if there is a long period in which the pathology can already be measured, but the brain is still fully functioning, then there is possibly the space to treat. And this is the link to the Alzheimer's disease drug development. I mean, of course, you probably all know, I mean, it's often on the journals. It's a very complicated topic. It has been, for over 20 years, there has been no medication that has been approved. The current medication, medications that are used and approved are symptomatic treatments, so they can, in a way, compensate the symptoms, but not really influence the disease evolution, no disease modifiers, apart from what happened last year. I'll make a brief comment on that. But in this field specifically, over the last decade, ultimately, there has been the increasing let's say, inclusion of PET, of biomarkers, and specifically molecular imaging biomarkers for the development of drug. First, as a biomarker for patient selection. I mentioned already that the clinical criteria are quite insufficient for an accurate diagnosis of the subject. So if the clinical trials that in the early 2000s here were based on clinical criteria ended up including up to 20 to 40% of subjects that didn't have the pathology ultimately. And of course, if you're testing a drug against amyloid, for example, having 20 to 30% participants that do not have amyloid is a major confounder in your results. But more recently, PET imaging has also been advocated and used as a surrogate endpoint, because ultimately we have markers for amyloid. The medications that have been mainly tested over the last 2 decades are immunotherapy against amyloid aggregates. So, of course, having a measure of amyloid in vivo can allow you to see target engagement. And some markers, and here I think specifically of tau PET, are clearly associated in clinical series to the cognitive outcome. So one could easily speculate that if you are able to remove or to reduce the accumulation of tau, you can favorably influence the disease progression. On the basis of this rationale, and this has been really a storm in the field, on the 7th of June 2021, aducanumab was approved by the Food and Drug Administration using the accelerated approval pathway. And the statement of FDA is really that this approval has been driven based on the surrogate endpoint of reduction of amyloid beta plaques in the brain, a hallmark of Alzheimer's disease. So what aducanumab clearly showed on the phase 1 results that were published. The phase 3 that I know of have not been published yet, is clearing of amyloid in the brains of the subjects treated. The large debate that took place in the community then over these last months is that this, let's say, benefit on, or this effect as measured on molecular imaging did not translate in those clinical series, at least not clearly enough, into clinical benefit. The clinical benefit was there in one of the 2 phase 3 studies that they performed, but it was quite small. There were side effects, of course, as every medication ultimately, and the whole process of the Food and Drug Administration approval has been, let's say, difficult at best. I mean, the Scientific Advisory Board advised against, then decision was taken to approve it anyway. The European Medicinal Agency decided late last year, December 2021, to not approve the drug. So we will see what is the evolution of this medication over the next years in Europe, or if novel studies are launched. I don't want to, let's say, take a position specifically on this aspect, but I think it's very important to highlight how, let's say, the molecular imaging part is taking importance into this type of decisions. And of course, novel studies and novel treatment developments will definitely take into account these type of measures, hopefully earlier in the disease, in the medication evolution. So not in phase 3, but possibly in phase 1, phase 2. This is what is already happening for another medication that is targeting Alzheimer's disease. This is another immunotherapy, another antibody, the donanemab. This is a New England publication, 2021. You see in this phase 2 results that the subjects treated with the drug have a decline, a worsening of memory, which is, let's say, milder as compared with what happens to placebo. Even if this phase 2 study is not yet, let's say, a phase that allows to draw conclusions on efficacy, it's encouraging to see this type of curves. And what's important for this medication is that the criteria for inclusion of the patients have been strictly determined on the results of amyloid and tau PET specifically. So patients were selected as being positive for amyloid on amyloid PET with a quantitative assessment. They were positive for tau, but in a way intermediate level. So having tau in the brain, but not too much. And what is even more interesting, these are only abstract reports from a conference last summer, but I find it very, let's say, very interesting to observe. You see that when you split this quantity of tau in the brain into 3/3, so the lower third, the middle third, and the upper third, and again, you have the same type of measure, so the worsening of cognitive decline over time. You see that the treated curve, which is the donanemab curve, so the green curve, is well differentiated from what happens in placebo, mainly in the lower third. So it's indeed the subjects which have amyloid, and who have present but very low tau that in a way determine the majority of the clinical effect that can be observed in these participants. So suggesting that really with the stratification of the 2 modality and of the amount of pathology in the brain, you can identify the responders. So of course, this is a perspective that I think will work in the near future. So I hope I convinced you that these molecular imaging biomarkers are useful to stratify patients, to identify the pathology, to identify it also in the early phases, and then use this information to select populations for drug trials, as we're not yet in the, let's say, treatment in larger scale phase. But of course, the molecular imaging biomarkers themselves have to be validated for this purpose. This is a work that we focused on over the last 10 years at least. Myself, in collaboration with a group of Professor Fusoni, who is the head of a memory center here, in the context of European and actually international collaboration. We tried to systematize in a way the informations that are there in the literature to then analyze the evidence which is there for molecular imaging markers in the same way that, let's say, in the same phase or in similar phases that are used for the development of a drug. But of course, with the drug, what you're looking for in the different phases of development from phase 1 to phase 4 is efficacy, so effectiveness on symptoms on a disease, which is an effect that you cannot expect from imaging markers. So from imaging markers, what you're looking for is precision in classification of the patients. And then also, let's say, given this precise information, let's say an indirect benefit in avoiding other conditions, in, let's say, ultimately improving also the quality of life of patients with an improved diagnostic process. That's why we adapted, let's say, the different phases of drug development, which ultimately results in a 5-phase, let's say, scheme, in which you have a first phase, in a first, let's say, status, investigating the analytical validity of the molecular marker. The phase 1 investigation would be phases in which you test, for example, your radiopharmaceutical for binding to in vitro the protein you're looking for, so tau aggregates, amyloid aggregates. A phase in which you would do preclinical studies, for example, preclinical PET can be very useful in this purpose to see, have first in vivo evidence that your molecular markers can actually reach the target and give satisfactory imaging, satisfactory kinetic. The phase 2 study has, let's say, as a central phase for development, the antemortem autopsy study. So studies in which you have your biomarker, your PET tracer, and you're able to see that indeed in vivo you can measure things that then can be confirmed at autopsy, which remains our gold standard. But then after having proven all this, so that your, for example, tau biomarker is accurately measuring tau pathology, you have also to show clinical validity in order to propose this tool in clinical practice, because you have to not only prove that you're able to measure tau, but also that when you're measuring tau, you're able to more accurately predict what will happen to patients, follow them up, etc. And then as, let's say, post-market authorization phase, you could imagine the implementation in which ultimately also cost consideration, of course, have to be taken into account, because as I mentioned rapidly, these are, of course, costly procedures. So ultimately, the advantage of this information has to be weighted in a way with the added value that is provided by these investigations. If we look at what's there for amyloid and tau PET, particularly because this is the topic I focused on today, I showed you at the beginning that the kinetic, in a way, the appearance of these markers has been sequential. So in 2004, the first clinical series are published for amyloid PET. The first pathological series are published in 2011. For tau PET, which is a more novel investigation method, the first clinical series appeared in 2013, and again, it's a bit less than 10 years later, 2020, that the first neuropathological series are published. Of course, these studies are complex and costly to run because they usually target end-of-life individuals that anyway, I mean, are followed up usually on average for 12 to 18 months because they're imaged and then followed up until death, to have then the possibility to compare the in vivo imaging with the postmortem results. You see here exemplary cases, often for the figures, I mean, let's say the most clear and the most positive cases are shown, but you see here, for example, the the binding in the cortex and the high immunohistochemistry signal that can be identified on cortex sections. And the same here for tau. So a positive image and then the presence of in the temporal and frontal gyrus of tau pathology. These studies have overall, with different methods, visual, quantitative, shown accuracy, performance above 88%. So in general, these tools are accurate in depicting in vivo in predicting or in identifying in vivo what would be identified then ultimately at neuropathology. But then, as I was mentioning, you also have to study, in a way, the benefit of having this information to take care of patients. And these studies need even larger populations. So for these studies, for example, we're not there yet for tau. For amyloid, there has been, over the last 2 to 3 years, 2 very large studies. One is a US study, which took, let's say, the real world approach. So what they did, these colleagues, the study is coordinated by Gil Rabinovich at UCSF, is to ultimately find an agreement for Medicare reimbursement for amyloid PET in study participants. They managed to include for these diagnostic study over 16,000 participants. And the threshold they set for a positive study was 30% of change in management. So what they did is the patient is included in the study. The clinician, the neurologist or geriatrician that takes care of the patient will establish his management on the basis of the information he or she has. Then the patient undergoes amyloid PET and the clinician is asked again, what is its plan and how it's implemented. And what was identified ultimately in this very large population was a change in management in, on average, 60% of the cases. So the majority of cases changed, for the majority of cases, the way the clinician deals with the patient, the recommendation, the drugs that are prescribed are changed by the information of the amyloid PET, which of course is a strong argument for its larger use. And here you see the publication, the results have been published in JAMA. The main change are an increased prescription in Alzheimer's drug in positive cases. Still in a landscape in which no disease-modifying drug is available or reimbursed, we could say, yet. In Europe, and you see specifically here the small dot, Geneva is actually the coordinating site. There is this AMIPAD consortium. I won't talk into details of what all AMIPAD consortium does, but it's an overall IMI study, so co-funded by the EU and the pharmaceutical companies also producing the traces, in which the information provided by amyloid PET tracer is investigated, and specifically in this diagnostic and patient management study, the diagnostic added value has been investigated. The design that we adopted here is a randomized control trial, smaller, let's say, scale, over 800 participants as compared with IDS, but much more controlled as of a design. And what we investigated is the 3 randomized arms of the study are: you either have an amyloid PET upfront, after, if let's say the clinical picture requires it, or you cannot have it for the first 8 months. And what we compare is the difference in, let's say, reaching a diagnosis that can be shared with a patient in 3 months. So in this case, it's really the diagnostic power of the drug, of the radiopharmaceutical that has been investigated. And the main aim of the study was to see a difference between these 2 arms, favoring an earlier access to amyloid PET. And the result has been obtained. The results are currently under dissemination and publication. You've seen that we have quite a large panel of tools then. I focused specifically for this presentation on amyloid PET and tau PET, but there are other tools also already in the molecular imaging world, we have FDG PET. The changes in glucose metabolism can be used as diagnostic sign for Alzheimer's disease or other conditions. There are other tools measuring specifically the dopaminergic pathway. So there are a number of elements. And one of the, of course, multiple studies have compared the different diagnostic value of the different tools, but ultimately the majority of these comparisons are comparisons between 2, maximum 3 modality. So it's impossible with, let's say, individual studies to have an answer for such a complex, let's say, landscape, and for specific recommendations of the sequence that have to be recommended in individual cases. So these diagnostic algorithms ultimately result so far, let's see if artificial intelligence helps in the near future, but so far result from expert opinion. So with the European Association of Nuclear Medicine, I'm active into it, as François has mentioned in the introduction. We've coordinated an international effort in order to define a diagnostic algorithm in Alzheimer's disease and other dementia. This was done as the usual, let's say, panel discussions with pro and cons argument of the different uses and the different steps that were discussed. And ultimately, we came up with this picture in which we all agree that the first step for the investigation of the patient should be structural imaging, and when possible, so when not contraindicated, MRI. And then based on the, let's say, specific clinical questions, you would start with amyloid markers or FDG or DAT-SPECT, which is the dopaminergic investigation I was referring to. And then based on these results, you would go on with second steps. But of course, and you see that in this picture, there's no tau PET yet. And why? I go back to the results that we discussed right now. When we see what is the results for tau PET tracers, we have convincing evidence of its analytical validity. As I said, in 2020, the antemortem autopsy results have been shown, but it's clinical validity. So the evidence that shows that applying it, and mainly in the let's say prodromal phase, so when no overt dementia is there yet, but the clinical signs are still initial, this has not been largely investigated yet. So the red color here refers, it's quite intuitive, to evidence that has not been accumulated yet. And so these results are weighted in a way before being able to suggest it into routine clinical practice. And another main limiting factor in using or in proposing these tools into diagnostic algorithms is their accessibility. And then here we go back a bit to the, let's say, availability of the cyclotron in a way, because if you see here, this is a survey that we performed in 2017, updated in 2019. It's been no major update in this sense, and probably there won't be any until the validation of a disease-modifying drug. You see that the standard tools, MR or FDG PET, which is a technique, let's say, cheaper and more routinely accessible, also because it's very relevant for oncological applications, can be performed in the majority of the European countries. But when we go to more targeted molecular imaging tools for the investigation of Alzheimer's disease, amyloid PET and tau PET. Amyloid PET is partly available in some countries, Switzerland included. Tau PET is not yet. So tau PET tracers, so far we've performed many here, over 200, but with the support of the Swiss National Science Foundation, there's no, let's say, a clinical access to this tool yet. And again, in the sense, having the possibility of, through a medical cyclotron that has also a research mission or research opportunities, this facilitates in a way the early access to these tools to clinical series. And I'm convinced that this is important because when you see also beyond Alzheimer's disease to the landscape of neurodegenerative conditions that are associated to dementia. Again, we go back to neuropathology because our gold standard is this. The different conditions, Alzheimer's disease is the most common, Lewy bodies dementia is the second most common, and then the spectrum of frontotemporal lobar degeneration ranks third within different clinical presentations. And what is characteristics of these forms are different pathological aggregates, amyloid and tau for Alzheimer's, alpha-synuclein aggregates for Lewy body dementia, tau, TDP43, and FUS for frontotemporal lobar degeneration. And then in the current understanding, as I was mentioning, the idea is that you have these pathological aggregates and then you have neurodegeneration that ultimately occurs. And in the current situation, the current markers that we have, we're able to measure amyloid PET, tau PET, mainly of Alzheimer's disease and partly in frontotemporal lobar degeneration conditions. We have PET FDG glucose metabolism measurement that is linked with neurodegeneration because, of course, neurodegenerating areas are areas that also reduce their consumption of glucose. But in a near future, I'm convinced that we'll be able to fill this figure even further with markers of synaptic density. These have already been published in 2 clinical series. There's none that I know of performed yet in Switzerland or available in Switzerland, but there are fairly good markers that measure SV2A as a synaptic molecule that is, of course, strongly linked with the neurodegeneration process. There is research, very active research into developing alpha-synuclein radiopharmaceuticals, alpha-synuclein specific radiopharmaceuticals. Here you see the result in a preclinical study with positive binding in a positive case. And for TDP43, we're still in the phase of molecule screening, but again, there is active research, big funding has been awarded in the last years. So we're optimistic that this will eventually happen clinically. So my proposal for conclusion would be that molecular imaging biomarkers have changed our understanding of dementia in clinical practice already for the diagnosis of these conditions, because we've really shifted from neuropathology to in vivo imaging and from care to demented person, that's the main objective, to the possibility of performing risk profiling in healthy, cognitively intact individuals. And the molecular imaging biomarker has also impacted a lot the development of treatments. The use of molecular imaging so far is has mainly targeted Alzheimer's disease as the most common condition, but is likely to extend in a parallel way to other dementia forms. There are many novel traces that are currently under development in different phases, some in already, let's say, fairly advanced phase as tau PET traces, others in the very initial phase of their development. And for this, access to a medical cyclotron is a key facilitating factor, and we'll try to, keep up with that. And with this, I would thank you for your attention and thank the funding sources for these projects.
Thank you, Valentina. Fantastic talk. Thank you so much for all this amazing information that you're sharing with the community. At this point, I'd like to welcome the audience to unmute themselves and ask questions.
I would like to ask the first question. It's a very nice talk. I'm Peter Nirmal Raj from EMPA in Zurich. And my question is regarding this is, you see, during your talk, you mentioned about the amyloid PET and the tau PETs, and you kept referring to them as the tau and the amyloid aggregates, right? Now, from the PET imaging, would it be possible to get the information on the polymorphic nature of these aggregates, especially at the earlier stages of the disease, let's say those with subjective cognitive decline or with mild cognitive impairment? Could you delineate within the information that you have as to, this is an amyloid PET scan with a person who has predominantly oligomeric aggregates, or is this a scan where it is predominantly with fibrillar aggregates? Could you distinguish between the polymorphic nature or the aggregate states of these proteins on the PET scan?
Of course, the nature of the aggregates or the presence of oligomers versus fibrils influences the binding. So I'm not really an expert into these chemical aspects, but from what I know, the amyloid PET tracers are mainly able to target fibrillar aggregates, and they're less sensitive in the oligomer forms. And the same is true for tau. Less mature forms of tau deposits are less visible. It's true that, and cannot be accurately measured. It's true that in the, let's say, in the field of Alzheimer's, with the main challenge clinically is that we are possibly too early on some respects. It depends on which side you look into it. But if you look at it clinically, so you have a patient with, for example, very early symptoms, some slight memory loss, or only worry in a way, but no memory loss, you measure them, you might have already amyloid and tau pathology, so already fairly advanced form of pathology, and you still might be able to follow them up for 3 to 5 years and have little decline. So in a way, from the let's say diagnostic perspective, these traces are considered sensitive enough. We're happy enough to see, let's say, these advanced form of pathology. Of course, the perspective changes if you're looking for the treatment aspect, because as we've seen, possibly for tau, the first phases of positivity are the only pathology that we can target. And when the positivity is slightly more advanced, the disease is already in a process that it's, in a way, it's self-maintaining and cannot be influenced anymore. So, but of course, and then these aspects, so the specificity for the different forms are aspects that change tracer by tracer. I didn't have time to go into details, but just to provide you an image, which is possibly of interest for, for the tau traces specifically, there is quite a lot of development now. You see possibly my figure now here. So you see there are these traces that are considered first generation tracer, the first that have been described with what I just mentioned. So a good affinity mainly for the most mature form. But there are second generation traces that seem more sensitive and so able possibly to pick up earlier disease. At the same time, for all these forms, for all these traces, the long process from first description, first clinical series, then studies to neuropathology has to be imagined. So their access to the larger clinical series is quite slow. There are a number of groups, Agneta Nordberg's group, for example, which is trying to do in silico modeling of the binding of these traces to the different aggregates. So, for example, for tau, different conformational structures exist in the tau forms that are associated to Alzheimer's or other forms of neurodegenerative conditions, for example, those associated with frontotemporal lobar degeneration. And so it's also well possible that these type of investigation help selecting, in a way, in the earliest phases, those radiopharmaceuticals that are most promising to be very sensitive to different forms and should go through the whole process.
Thank you very much.
Hello, Valentina.
Hello, Olivia.
Thank you very much for the presentation. It was really precise, but also really understandable for people not yet in the Alzheimer's disease field. I have a question regarding the early phases of the disease, because you said that amyloid and tau PETs were really useful to discover that the patient had the disease. So what are the criteria or the sign for a clinician to decide that the patient needs to do a PET scan before loss of memory or dementia?
Very good question, Olivia. So currently for amyloid, we have, there's been suggested in 2000 and let's say 11, 12, those that are called appropriate use criteria that were also used as inclusion criteria in the IDEAS study. And these are investigations that focus to patients that have already an objective memory impairment. So the patient comes to the memory center, tests are performed, the memory performance is lower than what's expected from, let's say, the age, and then they can undergo additional investigations. And this is also what is, for example, holds for the clinical use here in Switzerland. But the evidence in the earliest phases comes mainly from investigations, from studies in population studies. There have been large studies in cognitively impaired series. The largest have been done in Sweden, Australia, US. And in these very large populations, I mean, healthy populations are called and come in to have their imaging. And then these patients are followed up over time. And having to deal with the disease that progresses in the early phases quite slowly, so huge studies with thousands of people that are followed up over decades, we undoubtedly see that those that are positive for markers, positive for amyloid, and even more positive for amyloid and tau are those that decline, possibly slowly, but steadily over time, with a quite large individual variability. And the individual characteristics that influence the evolution are many. There's genetics, of course. There are, let's say, other conditions. So the fact that the patient has, for example, a high education, high occupational attainment. So there are a number of other factors that influence the speed and the specific individual clinical progression. This is one of the factors that renders clinical trials complex, because, for example, one of the discriminating factors possibly in the 2 phase 3 studies of aducanumab, one positive, one negative for the drug, is that the control population evolved differently in the 2 studies. And these are studies including overall 3,000 participants. So it's really a complex disease on these respects. So there are a number of other factors apart amyloid and tau that influence the evolution, but clearly amyloid and tau, either they are already signs of pathology or there are strong risk factors. There are different points of view of this, but they are determinants of higher risk progression over time.
Okay, thank you very much.
Thank you for a great talk. I appreciate it since we are a little bit linked with the Alzheimer's disease with our study. I have one question concerning the amyloid plaque detection, because you mentioned, okay, you partially answered my question, talking about the tau and the new markers and new radio markers for it. I have a question because sometimes this is not directly Alzheimer's disease. We have some other disease which are causing some symptoms of Alzheimer's disease. And after that, the deposition of the plaque, amyloid plaque, is not the standard extracellular deposition. We have the intracellular deposition. Are the markers which are right now, let's say on the market you have been talking about, able to detect intracellular deposition of the amyloid plaque, or this is still not, I would say, working?
It's a complex question. Per se, there's no limitation of the intracellular versus extracellular localization. So PET imaging is able to image intracellular targets because, I mean, depends on the characteristics of the tracer. The tracers that have brain penetrance are lipophilic tracers, so they can also come across the cellular membrane up to a certain point. Of course, the main limitation for imaging is, in a way, the abundance of the target. So if you have, and this is one of the elements that have rendered the fact that tau tracer came later than amyloid tracer, because, I mean, it's a less abundant target and will probably be an influencing factor also for TDP-43, for example. The current amyloid markers image, they're not specific for, as you said, Alzheimer's disease. So there are plaques also in other conditions. For the differential composition or contribution of intracellular and extracellular amyloid, I wouldn't be able to answer you competently, but we can look at it together.
And Valerio, you have a question.
Yes, I have a question. So thanks, Valentina, for the talk. It was really nice. I left the field of Alzheimer's back for my PhD, so it was really nice to see how it evolved, and it evolved quite a lot, I have to say. I had a question because your whole talk was, of course, focused on the amyloid and PET tracers and tau PET tracers. But then when you show the picture of the schematic of your consensus paper of really what to do with a patient, the first thing you still do is structural imaging. Could you tell us a little bit more of why you do that, or where the structural deficit, which I would assume that maybe hippocampal structural damages that you would look for, where are they positioned in the? Are they a very early marker, or they come later?
Yeah, so very good question. So in clinical practice, MR is there upfront. And in a way, it was already, I didn't mention it specifically, but in the 1984 McCann criteria, even if MR was not specifically measured, because possibly, I mean, MR was little or no available at the time, but the idea of having a morphological imaging that can exclude the presence of other, what one call secondary forms of dementia. So for example, a lot of vascular pathology, multiple infarcts, a tumor that could mimic this clinical situation we would observe has to be done.
Okay.
Then, of course, MR is not limited to this. I mean, MR has a number of, very relevant markers, and one of, as you accurately mentioned, that is used clinically is the volume of the hippocampus. The hippocampus is one of the main, let's say, target structure that is involved. It's directly linked with the memory presentation of Alzheimer's disease, and it can be accurately measured. This is, let's say, is at the same level of the changes of glucose metabolism. So it's a sign of neurodegeneration. It's considered as such, and so it's considered a downstream event from amyloid and tau accumulation. You can have amyloid and tau in the brain and still no atrophy, but the clinical symptoms usually occur when atrophy is there. So in the diagnostic algorithm, what is suggested includes also this information. And clearly, if you have a patient in which the clinical presentation is typical and typical atrophy is observed, and the patient is not eligible for a specific medication, is above 85, for example, which is usually considered a threshold in which, no much space for intervention is there. Clinicians might feel confident enough to say that there are sufficient elements for a diagnosis of probable Alzheimer's disease. That, let's say, symptomatic drugs can be administered and the algorithm stops there. But if you have a patient with a typical clinical presentation, 60, still working, and some mesial temporal atrophy, you still might not be, let's say, confident enough to conclude that's Alzheimer's, full stop, because there are other conditions, for example, TDP-43 pathology of the mesial temporal region. This is not typical for young, but let's just forget that for a second. That could have, let's say, a mimic from the, let's say, clinical point of view, from the imaging point of view, and still no pathological hallmark of Alzheimer's disease. And depending on what you want to do, if you want to start, for example, inclusion in a clinical trial, the sign of a non-specific neurodegeneration wouldn't be enough to go further.
Okay. And maybe another related question. So when you, for example, or is there a study that showed that clearly, if you have, for example, the same subjects, the same patients, you have amyloid PET and tau PET markers, whether these two, they tell you slightly different, they give you a different information regarding the evolution of the disorder, and they don't tell you basically the same thing. I could imagine that if they are, if in the end they're stratifying so they're used with the same criteria for stratifying the patient, but if they tell you the same type of information, that you could choose either one of the two, or they are complementary. So somehow, if you use both, your accuracy really significantly increases.
There are a number of studies using the information and showing that the positivity of both amyloid and tau is, of course, associated with a higher risk of progression, but the positivity of tau alone could possibly give the same answer. So I agree with you. The majority of the studies didn't do this, let's say, didn't have to do this choice, because I don't think that there are yet A+, T+ studies longitudinal with imaging. The majority are based on cerebrospinal fluid. I gave you a very PET-centered view of the situation. Of course, amyloid and tau can also be measured in the cerebrospinal fluid. And if you make a lumbar puncture, then you have both measures, and then you do not need to choose in a way. But it's true that the way, the measure in the cerebrospinal fluid is sort of an indirect measure, while with imaging, you can see its accumulation in the brain. And in that case, of course, you have to choose because, also for cost consideration. And the current knowledge for tau PET positivity is that when tau PET can be detected beyond the mesial temporal lobe, amyloid can be, in a way, assumed automatically. So amyloid is almost always there. So I agree with you. This is one of the topics we're working on, but I think we could show that my personal view on that diagnostic algorithm would be to put tau PET, and based on its results, stop the algorithm quite soon. Okay. In the earliest phases. But of course, before being able to suggest such a diagnostic algorithm, tau PET should be more widely accessible than it is now.
Thank you.
Hi, Valentina. Thanks for this very comprehensive presentation for people like me that I'm not so familiar with this type of imaging. But you didn't comment so much at the end as regards the imaging itself and the automated image analysis. Which are the major drawbacks, if any, because maybe you are happy already with this, if I understood properly, is kind of a quantitative imaging. You can compare it across centers, across patients. Maybe there is some normalization, but you are doing measures, I guess, overall of the how much is highlighted or difference from the normal. But me, I see blobs, I see very fuzzy images. So can you comment on if there is any aspect that should still be improved in terms of image, spatial resolution, and other artifacts, please?
Very good question. So of course, the analysis of images, again, going back, let's say the certification or the validation process implies also the way it's measured. So that's why the measuring is so far been kept quite simple. So it's visual assessment of these blobs plus quantitative measurement of let's say global load in the different regions. Voxel-wise analysis are often done and usually, let's say, reliable. The resolution of the technique per se is so far limited by the resolution of the tomographs and by the physical process. So the resolution of the tomograph is now in the order of the 4 to 5 millimeters. This is the resolution of the PET. So it's not particularly high as compared with MR. There is a lot of research in the direction of improving this. The limiting factor in a way is the ability of the whole chain of event. PET imaging is built from the detection of the two photons that are emitted by a positron annihilation in the tissue. The current detectors and detecting systems allows a resolution of up to 4 or 5 millimeters. This has been reduced, I'd say, to 3 millimeters in brain-dedicated systems. There is the HRRT. It's a fairly old system that has been suggested for that. This resolution is, again, the topic of a number of investigations. There is, for example, a number of projects here, actually also in collaboration with CERN. Of course, they're experts in detecting. And so I hope we'll be able to soon see tomographs that performs better, that go in the millimetric range. And that will, of course, have also an impact on what kind of pathology we can measure, hopefully having more sensitivity for earlier types of accumulation. So there is I think it's an exciting phase for this type of development. Another, let's say, front in which there is a, let's say, technical development is the design of the systems. So far, the majority of the systems are fairly large bore all body systems. Because in order to make the PET systems sustainable in a way, you want to image not only brains, but also the rest of the body, because there are a lot of indications for the rest of the body, chiefly oncology. But of course, you could have possibly in a way, a cheaper or a good compromise in having organ dedicated systems. And so there are now finally commercial products that focus specifically on imaging the brain. So with these, improved resolution can be expected as well. So.
Ileana, would you like to ask your question?
Yes, thank you. Hi, very nice presentation, Valentina. I agree, very clear for pretty much all levels of knowledge on Alzheimer's disease in the audience. I have a question. I actually want more your opinion, because you discussed a lot about amyloid and tau, and we don't know which comes first, etc. There's a lot of controversy, but what if we also throw in impaired glucose metabolism in the mix? Because some people have actually argued that it comes in earlier than we suspect, and even labeled Alzheimer's as some kind of type 3 diabetes, so diabetes of the brain. So I was wondering if what is your take on the fact that it may come from impaired glucose consumption earlier than we can even detect with FDG PET, for example?
Yeah, I mean, of course, this is a very active research field, and this is a theory that has been out for a while and that is revamping recently. Of course, I mean, with FDG PET, we have a measure for that. So it's in a way, it would be very good to see if we can detect anything earlier. But as you say, I mean, it could be an alteration of the pathology that is in a way non-detectable yet on our images. So for the specific, let's say, I don't have a specific opinion on this causal process versus others. Another, let's say, theory that I find quite convincing, I saw very nice data presented by Professor Henniker, for example, a year ago or so, is the involvement of microglia. You know that, I mean, and again, in that space, I mean, if it's microglial activation following pathological aggregates, or the other way around, the microglial dysfunction causing pathological aggregates, and the whole disease cascade is not known yet. It's true that in order to know the pathological cascade, disease, let's say drugs able to positively influence the pathological cascade are the missing link of the disease yet. I mean, there are a number of models, preclinical models that have been efficiently treated with a number of strategies, blocking amyloid, blocking tau, influencing microglia or glucose metabolism, but so far in a clinical translation, we're still quite far from this. What's more advanced is the amyloid theory, strengths and weaknesses. It's true that what we seem to see, even if with noise and possibly in a too late phase on medication that influence amyloid load, is, in my opinion, quite promising. So I would be happy. I think that that's still, I mean, not out of the. I wouldn't bar the amyloid hypothesis right away. I think that it has some solid into it. It also comes from, let's say, that from the. Observation of genetic influences, of course. I mean, the monogene forms of Alzheimer's are a strong argument in favor of that. Then it's clear that Alzheimer's disease, sporadic Alzheimer's disease observed in aged individual is probably not a mono-process, mono-chain disease. So a number of factors come into play. And so, yeah, when glucose metabolism is possibly one of the driver, it's something we'll find out.
And regarding, so you need to find a target for therapy, which I think is really a very important question. So do you think that eliminating tau neurotangles is actually more promising than eliminating amyloid plaques, which have nonetheless shown only moderate results in clinical trials in terms of improving the person's cognitive performance?
There have been in studies, I mean, same approach, antibodies against tau that have been tested, and so far with results that are as or even more disappointing than when targeting amyloid. So, I mean, but again, possibly the type of forms, possibly the phase in which it's applied. I'd say that it's difficult to have a final answer for that. in my opinion, I mean, a lot of multimodal investigations in the earliest phase of drug development is the way forward to find effective treatments.
Okay, thanks very much.
Okay, any other questions or comments or advice?
I have a curiosity. Hi, I'm Christina. Thank you for the presentation. It was really nice. Maybe you answered it and I just missed it. The two traces, so you showed three tracers at the beginning of the talk. It was the FDG-PET and the tau and the amyloids. Are they available to be transported in Lausanne or somewhere else? Or
not only available to be transported, but routinely performed. I see that John Prior is in the list, but I don't know if he's connected, but still the amyloid and FDG are clinical tools, and they're, I mean, routinely performed and done in Lausanne, and tau is done in Lausanne within a collaboration with us and Zurich. Okay. But I see that John has appeared, so I'll let him answer.
Yeah.
That's true. Thanks to you, we are able to perform tau imaging and amyloid imaging also for the clinics. Tau is still a research project that we're gratefully to collaborate with you in Geneva.
Okay, because yeah, I see Bernard here. We have the preclinical PET at EPFL, and we do have DigiPET who comes from Geneva. But I don't know if you have the possibility to do something else than FDG-PET. So I was just wondering because, yeah.
I'll disclose the source as well of these traces. So both amyloid and the tau are produced in two different cyclotrons. So the amyloid tracer is produced in the cyclotron at the USZ, at the University Hospital. And the tau tracer is produced at the PSI cyclotron. Just to provide you, let's say, a general Swiss map of the cyclotrons, we have the cyclotron in Geneva, the cyclotron in Bern, which does mainly, let's say, commercial products. And then there are three currently cyclotrons in Zurich, two at the University Hospital and one at the PSI. The University Hospitals has, let's say, clinical production, and the PSI mainly for, I mean, research project and not for clinical use. So we wouldn't be able to ship tau or amyloid tracer. We receive it. We, I mean, distributed it also. We agreed in order to have a distribution in Lausanne within a research agreement. What we are planning for preclinical imaging, because we're planning the same here in Geneva, and of course would be very nice to do it multi-site then, is to, of course, the main restriction then is costs, because the costs for a single dose are high, even within research agreement. As I showed you, I mean, it's a synthesis per subject, more or less, so per a few subjects. When we do amyloid PET, we do usually 5 here, and the shipment is probably for, I mean, maximum 20 overall Switzerland. So it's a synthesis that has a lot of costs. So it's difficult to imagine to reserve a dose for animals. Yeah. But what we are planning to do is to use the residuals. Of course, the activity that is needed for humans is a bit more than the activity that is needed for animal. So we will explore the feasibility of using the residuals for preclinical experience, and possibly the same can be discussed in Lausanne.
Yeah, because it's difficult. So there wouldn't be any. So the only limitation is the transport of animal and the animals and the authorization to have them on the same site, you know, to do the PET or the PET in Geneva. So we would need to change everything. And now with the new election and everything, I'm a little bit,
Yeah, sure. Yeah, that's another aspect as well. Yeah. I mean, it goes back to my, let's say, my claim for the relevance of having a cyclotron in the region that can be available for thinking also these preclinical developments. Let's say the access to these techniques, if it's only in the hands of commercial companies that have to, let's say, develop a tracer to mature enough to be able to commercialize it and bill it and have it reimbursed is a strong limitation. So have some flexibilities in being able to produce tracers, not only for commercial purposes, is of course important, but of course it has costs that then wouldn't be, let's say, for companies, but would be for institutions, which is an issue as well. In Geneva, we're still working. I mean, again, Professor Prior hears this story since a number of years, so we should work on our credibility, but we're producing now a tracer for microglial activation for which is PBR111, a tracer for TSPO as a target. So that's another target that is, relevant for the field, and that we'll try to disclose further when we're, ready to ship it in a regular way. And of course, we produce FDG.
Okay, thank you very much.
Okay, Bernard, this is the last question because I have a lot of news to tell you.
Thank you. Yeah, it will be, well, the question at least should be rather short. So I'm Bernard Lance. I'm interested in working on isotopic labeling studies, both on the MR side and the PET side. And actually, I'm quite interested in analyzing as quantitatively as possible the kinetics of labeling of those traces. So going in the direction of what Mary was asking in terms of spatial resolution, I was wondering whether you looked at the kinetics and whether that could improve the sensitivity of the measurements you do with your tracers. So if you see different kinetic profiles in those diseases.
Yeah, absolutely. So the majority of, let's say, what's validated clinically is static imaging at a given time point. But of course, in the, let's say, radio tracer development and then first clinical series, dynamic imaging is performed in order to see the dynamic of accumulation. And then in the AMIPED study, we tried to, let's say, make this dynamic information available at a larger scale, because the problem there is that there are a few tens of patients that are imaged dynamically, usually, showing that if you take also the information of the tracer arrival and earliest phase distribution, you have additional information as compared with just measuring it at equilibrium, if equilibrium is reached. But then this is done in a few studies in research centers and then disappeared. And when you use it clinically at large scale, you do only the simple and, let's say, less precise way of doing it. For a number of considerations, including costs, because of course, if you do a dynamic imaging of 1 hour and a half, you take much more space on these on our machines than doing a 20-minute scan. So the compromise that has been developed, and I find it convincing in the AMIPED consortium, is, let's say, two-phase imaging. So first 30 minutes in order to have the, let's say, the peak of the access of the tracer to the brain, and then the first phase of reduction, then 1 hour pause, and then 20 minutes. This is tolerable from the patient point of view. It's twice 20 minutes. And at the same time, it's sufficient to have accurate measures of tracer distribution. So it's the Amsterdam site, which is leading the development in this sense, Professor Lammertsma and then followers. And then with this type of information, you can, for example, extract from one acquisition already two information, the amyloid load plus the perfusion of the brain, because these traces pass rapidly in the brain, and then their first phase of distribution is an image, and then can be derived more precisely from the whole dynamic, is a measure of brain perfusion. So there is added value, because then also in amyloid negative cases, you could have information on brain perfusion that can guide you investigating the non-amyloid related forms of dementia.
Thank you very much.
Okay, thank you so much to Valentina, first of all, for this stimulating talk that generated so many questions and ideas, and I look forward to many collaborations together all over Switzerland. And this is a really great start to 2022. And Valentina is now the new section head, so please help me to welcome her and work together all in this amazing field.